I want to talk to you a little bit about some mechanistic insights about inflammation and resistance specifically. Can you just briefly walk us through the specific inflammatory signaling pathways most implicated in promoting therapy resistance?
Yes, so one pathway that really we study in the lab and has been highlighted by another group as well related to venetoclax resistance is interferon gamma signaling. So interferon gamma response inside malignant cells starts from the interferon gamma receptor, then causes autophosphorylation of JAK2, which leads to accumulation of molecules called STAT1, STAT3, and STAT5 that then they're getting phosphorylated and moved to the nucleus and regulate the expression of a number of downstream targets. Some of them are interferon-related genes, IRFs or ISG, and also a number of receptors, eventually cells, proteins that are expressed in the cell surface, such as ICAM1. So it looks like the majority of those genes, their expression is linked to venetoclax resistance. And that's what we are really interested in understanding. One thing that has attracted our attention, this is what we published recently, is that deletion of P53, which is a protein that is critical for the repair of DNA damage, cell cycle arrest, even apoptosis as a result of DNA damage stimuli. And the loss of P53 really makes acute myeloid leukemia and myelodysplastic syndrome really, really hard to treat because simply the cells cannot die. They are very resistant to most of the apoptotic mechanisms or other mechanisms of cell death that we use. We utilize giving venetoclax or chemotherapy. Then the loss of P53 we saw that causes a direct upregulation of interferon gamma signalings, like without any implication of the bone or marrow microenvironment. So it seems that there is a cell intrinsic phenomenon linked to P53. Obviously, we need to further confirm that in a number of models and understand it, but I think this could be an important contributor to venetoclax resistance that we see in these malignancies.
Interesting. What are some of the key post-translational modifications that appear to regulate these inflammatory cascades, and how do they influence cell survival under therapeutic pressure?
So we know that phosphorylation and ubiquitination are all key important post-transcriptional modification for a number of those genes. We have studied a little more phosphorylation of STAT1 in the context of activation of molecules such as JAK1 and JAK2, and all this obviously creates a much more complex concept because we cannot rely on RNA data and transcriptional data or single cell RNA transcriptomics. We need to understand more proteomics and understand also the regulation at the protein level of some of these pathways.
Mhm.